Actually, what I'm going to talk to you about today very much was inspired by Hud and following his vision, trying to see ways in which we may be able to impact some rare diseases, coming from the perspective really as a drug hunter. So I've spent 15 years in a pharmaceutical company and run the Institute's translational operation, which I'll tell you a little bit about. But we come at this with sort of rather a pragmatic viewpoint, which is wonderful science going on, but how about just throw some compounds onto patient cells and see if you can change the dysfunction. So I'm going to tell you a little bit about that. It's somewhat controversial. Some people say shouldn't really be going that way. Others say, hey, that's kind of a cool idea. Give me some proof of concept it could work. So we've got a bit of a mixed audience anyway. I've kind of got some slides that are sort of dumbing it down a bit and others that dive back into the science again. So hopefully you can all get something out of the presentation. So after this morning's, the keynote speaker, I think, I have to revise. It's now over 6,000, 6 or 7 or 8,000, whatever. It's increasingly clear, though, that as the sequencing takes off, the question is moving not so much from diagnosis, that the traditional problem of patients who don't develop appropriately, moving forward, coming to physicians who have to try and guess what's wrong or figure out what's wrong through the traditional methods of taking blood or tissue, doing biopsy. That's starting to get increasingly replaced by going to sequencing. And I would think we'd all predict that in the next 10, 20 years, it's all going to go the other way, which is the sequencing will somewhat lead the issue up front. So now the problem is going to be moving to, what about the treatment options? And I think it is certainly read that for the 6,000 or so rare diseases, maybe just for a couple of hundred of them, there are actually treatment options. So that's not a very good ratio. And good news, I know what's wrong with you. The bad news is I have nothing really to treat you with. I think that's kind of what we are all a bit frustrated with. So is there a way to try and think about accelerating this process by which we might be able to come up with some treatment options? And if you heard of some of the talks today this morning, we're sort of reusing either natural products, things we eat. You think, well, that's even more aggravating. Those things are already there. We just haven't figured out which patient to match them with. And so the other side that I think is important here to realize is that many of the drugs that are already approved out there on the market are kind of what we call dirty drugs. They don't work one way. They actually have multiple mechanisms of action. And so that's actually to our advantage in trying to look how to reuse those specifically for diseases. And so a couple of things have happened over the last 5 years. Not only has the sequencing changing the equation to move from diagnostic to the search for treatment options. The next thing that's moving along is our ability to be able to use stem cell technology to take from an individual their cells, generate stem cells, and then any cell pretty much of the human body starting to get to the stage where we can do that. And I'm sure many of you have been following this field within the Sanford Burnham Research Institute. We're very fortunate to have about 100 or so of the scientists are working, advancing the field of stem cell biology, patient cells. This is where you take skin cells or blood cells, you reverse program them to become stem cells, and then from that you reprogram those stem cells and differentiate them to make many different types of the body. Now what's so exciting about that, of course, is that it starts to allows us to get to cells, an unlimited number of cells. They have the genetics of that individual. You remember, all of us are different. So they have the exact genetics of that individual. And if those cells were to demonstrate a defect, a functional defect, what about just screening those? So that would change the paradigm almost entirely to this type of thinking, which is where we would be able to take blood or skin cells. If we can find a dysfunction in those cells straight away, that may be something we could screen, looking for drugs that would reverse that dysfunction. So what do I mean by dysfunction? Well, you heard about some of the things today, but straightforward with things, if a muscle cell, a cardiomyocyte, doesn't beat quite correctly, you can actually measure that with a microscope, and you can actually measure that defect. If a cell doesn't stick, doesn't migrate properly, doesn't adhere properly, you can measure that dysfunction. So we have within the Institute some of the most powerful microscopy, high throughput microscopy setups that money can buy, right? It's the most sophisticated, quantitative cellular imaging and functional analysis, either of live cells or of fixed cells, to give us that quantitative endpoint that can determine whether that patient's cells are normal or whether they're dysfunctional, and particularly where that function connects into what is, we believe, wrong clinically with them. That's a very powerful thing to start thinking of screening. So this starts to allow us to get to cells that are an unlimited supply of a patient's cells, not from some mouse. This is actually a patient, right? Patient cells, we can then amplify them, make, say, the cell type that has the dysfunction. We then, what we call screen them, and I'll tell you a little bit about what we mean by screening, which is where we develop assays to be able to look at that function and then look for repositioning existing drugs that could reverse that function, ameliorate that function. So we're not necessarily looking to fix the problem. We're looking to ameliorate the problem. Now, why do we think we could possibly be successful there? Well, I think as you heard earlier, two individuals can have almost identical genetic defect, but the clinical manifestation of that disease can be quite different. That tells you that there are compensatory mechanisms outside of simply fixing with, say, gene therapy, the exact problem you have. If you could tweak these things up, make it so rather than being a rather severe disease, it's a much milder disease, well, I call that some level of success. It may not be a home run, it may not be the cure, but we're making an impact. I mean, it's an immediate impact you can think of. So this is looking for actually being able to provide back to a physician taking care of a patient possible treatment options they may want to think about. But this dysfunction that this patient's cells had, when we put compound X on those cells, improve that dysfunctional endpoint, all right? So that's the thinking, right? So it's pretty, it's not very intellectual, actually. It's, you take the cells, look at the dysfunction, and say, do any of the things that we have out there, can any of them change, move the needle? Now, the disappointing thing is that we don't actually have that many drugs. 2,000 or 3,000 drugs that are actually approved out there. That's not many. So that's, there are some, we could take natural products, metabolites, throw in a few more thousand of those. Still not a very, normally when we screen in the drug industry, we screen 500,000 compounds. So, you know, you'd kind of say, nice idea, but is it really going to work? So, sort of goaded by HUD to a certain extent that this is maybe not going to work, but, you know, I said, well, I reminded him that his Manos experiment worked in Fibroblasts. So he says, oh, okay. So, so what about if we try this on some sort of scale? So the institute, as I say, fortunately at Sanford Burnham, we have many of the components to try and pull this together. So disease in a dish, you may have heard the term, those who haven't, it's not something in a student's refrigerator. This is, right, this is the idea of a patient's cells that are dysfunctional, being able to measure that dysfunction, the component that's wrong, and being able to see that, looking at those cells really representing a mini human, right? It's like a little mini representation of a model of them that's wrong. Looking to use those. We're going to use a combination of the chemical libraries we have, mainly the approved drugs, but also some metabolites where we can. What we call high content screen. This is microscopy-based screening, using highly quantitative technology that can determine the degree of this function from one cell type to another and see whether we can, with using a lot of statistics and high power pattern recognition technology to determine that dysfunction, trying to find things that will move the needle and move it back to more the normal state. And we're also very fortunate to be able to make stem cells as well, and then from patients' cells, which then can be differentiated. And my partner in crime, Ann Bang, and I see Evan's just arrived, another of the stem cell experts here, are driving this side of the equation. So this is kind of bringing a number of components together. And specifically, the component we're really leveraging is the Institute's translational capability, which I'm not going to go much into, but we have tremendous robotics and liquid handling and big robots that kind of can move, do these screens and have the technology to be able to really quantitate in a dish what is going on. This is called HCS, high content screening. And what you're really doing is you're using pattern recognition to identify something that's the component that's wrong, right? And the higher up in the order of wrong, rather than just looking for the absence of the protein, say it's a hypermorph, in other words, not too much of the protein's present, we're looking for the thing that it functionally contributes to that. So in the case I'm going to show you a little later, it's where the dysfunction leads to the lack of adhesion of the cells. So I can then measure with a screen how well do those cells adhere, or how well do they bind to their partner. And I'll show you an example of that. And it takes a tremendous amount of infrastructure to do this, and we have that all set up. So that's the one side. Another side is the stem cell world. And really, at the end of the day, this is the three things you're bringing together. You're bringing together the drug collection, the patient-derived cells, and the technology, the assay, the screen. And of all of this lot, it's the assay that really matters, right? What are you actually going to measure? And how reproducibly can you measure it? And how do the patients look? How do their cells look? And do they look dysfunctional? And to what extent can you measure that with precision? So I arrived at the Institute a couple of years ago and went around talking to many of the PIs, particularly in the Children's Center here. And we started talking about, well, which would be a suitable disease? Well, let's try and get a proof of concept here. I mean, it sounds a good idea, but can you actually make this, bring it into practice? So as we've heard a number of diseases, most of the ones you heard today would qualify in terms of a disease with a known genetic basis. We're not, we need to know exactly, these are the proteins that are malfunctional. We prefer to be working on something what we call hypermorphic, not completely missing the protein, but maybe it's got 5% of the amount of protein. So that would allow more compensatory things to be able to kick in. It would result in a dysfunction of cells that can be measured with existing reagents. So we don't want to start from scratch. We want to be getting going quickly here. And we want to do what we call a black box screen, a non-intellectual screen. So basically, you have the cells, they have a dysfunction, you put compounds on, and we don't care how they work. Well, we will later. We'll go try and figure it out later. But right now, at this time, can I change the endpoint? Can I actually alter that dysfunction and make it closer to normal? And really, quite importantly, that you'd really like that thing that you were measuring could be considered close to a clinical biomarker. Something that's sort of a surrogate endpoint, something that if you move that endpoint, clinicians would pay attention and say, hey, that's pretty cool. If you can move that, it may not be a physiological measurement, but it may be a biomarker or something we can measure that people would believe that was significant to improving the disease. And of course, we need access to the patients and those samples and people who want to contribute in this area. So we chose in this area, it's called the Dystroglycanopathies. And you've heard today already, the previous speaker was talking primarily about N-linked sugars. This is a very special sugar modification that's made particularly on alpha-dystroglycan. It's called an O-monosyl sugar. And the details don't matter too much. But just as in the last talk, there's a series of enzymes here that decorate this sugar complex. And in so doing, make a high affinity binding site for laminin. So why is that important, and why do you need to have that? Well, really what that's doing is connecting the cell, which is shown here. Whoa, I'm not very good with my mouse. Here, to the extracellular matrix. So Duchenne muscular dystrophy and Becker muscular dystrophy have problems here in deficiencies in dystrophin. This is inside the cell, inside the muscle cell in particular. Problem in the dystroglycanopathies is you don't make a specific sugar here, which is modifying alpha-dystroglycan, which generates a high affinity binding site for laminin. And in so doing, if you don't have that high affinity binding, you don't connect properly, and if you don't connect properly, then you are not actually there's not rigidity and there's not connectivity, and so the cells become fragile, and all sorts of bad stuff happens downstream of that in terms of muscle function. So the thing that's for defects in any of these particular proteins here all result in a lack of binding or a reduction in binding between the extracellular matrix and the muscle cell here, due to the absence or the reduction in the amount of that O-monosyl sugar. So the question would be, well, if I took patient cells, could I measure that? Could I actually see a reduction in the binding of laminin? Because if I could, that's pretty close to what I was talking about, an assay that will allow us to screen for drugs that would improve the amount of laminin binding. All right, so that's kind of the thinking here. So what we're really trying to do, though, in the case of the dystroglycanopathy, where you don't have a high affinity binding site between alpha-dystroglycan because the sugar is not there, to laminin, we're trying to find things that would take us from this state to the other state. And so what we're really looking for is we're going to set a screen up of existing drugs or compounds which are bioactive compounds or metabolites, and we're going to do a black box screen trying to find things that will take us from the dystroglycanopathy state to the unaffected state. Moving is more in that direction. And it can happen by, you can imagine a number of ways it might happen. Right now, it doesn't matter too much why it happens. We can dig into that later. First is, can I move the endpoint? Can I move the needle? Can I change it back closer to the normal state? So here, we've taken patient cells here, and what you're seeing in blue is, this is a picture down the microscope, blown up, amplified many times. The blue dot is wherever we have a cell nucleus. And shown in red is where we stain the cells with laminin. And we've made the laminin red, right? So now you can see a normal person's cells, they bind laminin just kind of fine. And then we're going to use the microscope, and the high content microscope, to quantitate this. So it's going to quantitate the precise amount of redness, so the amount of laminin binding. How much laminin binding does each of the cells, each one have? And this staining technique took us a long time to actually put together. It was helped actually with a card-carrying glycobiologist here, who's also in the center here, Minoru Fukuda, helped us with the reagents of how to do this. It's not as easy as it kind of was popped out to be. But in doing that, you can then say, all right, so I can then quantitate and say, what about if I take a family in which I've got an affected individual who I know has got a mutation in one of those enzymes? How do they look? So here, hopefully you can see this. It's a little bright, the lights, but hopefully you can see. I think it's fairly, you don't hardly need a high content microscope to actually see the difference here. So here are here's a family, a father, mother, and an unaffected brother. It's a twin brother, but obviously not an identical twin brother, and an affected brother. And you can see very clearly, I think, in their cells, that the amount of laminin binding fits very closely to the genetics, right? The genetics is that the affected brother has mutations in this case FKRP, which doesn't matter, it's one of those enzymes that adds the sugar. And so now you can start thinking, well, I have a screen then to a certain extent, right? So here's, if you're following this, I've made a sort of cartoon version of this, right? So here's a normal cell from an individual that normally adds on their alpha-dystroglycan, has the O-link sugar, right? Whereas the patient cells have very little. I've shown none, but actually they have a little, to be honest with you. And then if the normal cells bind laminin. So if we actually look in the dish, the reason you're seeing all those colors, right? The blue is the nucleus and the red dots are the laminin, right? So now you can sort of see that we have a screen. So this is kind of what we mean by a screen. What we do is we take a plate that has 384 little wells in it, and each of those wells we put the cells that don't bind laminin. They're the patient cells. They're not able to bind laminin because they have that deficiency. We then take the active ingredient from a collection of drugs and put in each of the wells the drugs and say, can I actually move the needle? Well, exciting to say the answer was yes, right? And surprisingly, in quite a few, we have found 10 compounds that would actually move the needle. So what I'm showing you down here are, they're actually pseudo-colored. These are the 384 well plates. And where you see a red dot means that I now have red binding, right? And I've got duplicates here. So the top 4 and the bottom 4 are the same plates, but in duplicate. And I think you can see, just to pick on one, this particular well here lit up red because it now, because of the presence of that compound, has increased laminin binding in that particular cell, I mean that well. And you can see here, as these were duplicates, you become a believer when you do it twice. You can see there that it did that. So that was pretty encouraging. And we screened, in this particular case, about 5,000 compounds in duplicate. We screened the library of pharmaceutically active compounds, a drug collection for approved FDA drugs, EU drugs, quite an interesting collection here of called a green pharma collection, which are pure natural products. And of those, we found what we call 10 hits. So 10 compounds from that collection of 5,000 were able to move the needle. So I'm going to show you the most spectacular one of those, which was pretty spectacular. I was impressed, I have to say. So here you go from the unaffected brother, the affected brother, and then the infected brother give, in this case, 5 micromolar of the compound. So it's not like 100 micromolar, it's 5 micromolar, right? So, what you can see here, I hope, is that you've gone from really no laminin binding to pretty impressive laminin binding. So that was pretty encouraging. Now, the first you'll say, well, how does it all work? We're not getting to how it all works right now. We're going to see, is this a reproducible thing? Is this something that we can do on multiple patients, et cetera, et cetera? So, and would that compound, it is actually a known drug. Is it within the clinical range at which that drug is actually used? So the first experiment to address that really is to have a look over a titration here with no compound. As you increase it, you can actually do the pharmacologist, you like to get these curves that tells you the IC50, so that, approximately what sort of concentration do you need. So we've got an EC50 here, about 1.9 micromolar. That's within the clinical range this particular compound's used. So that's encouraging, right? So does it just work on one patient? So Anna's gone on to collect samples, and a number of the advocacy groups have really helped us here, collect more samples of patient cells here. And if you focus on the top panel rather than the bottom panel, so the top panel is actually quite interesting in itself. So all of these individuals, there's 5 patients shown up here. There is a degree of severity of the disease, starting off on the left-hand side with the most severe form of the disease, which has the name muscle eye brain. So it affects all of those organs badly. And going over to the far side over here, which is more of a limb girdle muscular dystrophy, a less severe form where the patient's really starting to have problems at about 15, 16, 20 years old, that sort of a. And what I hope you can see just in the top panel alone, that the degree of staining, it's not, you need the quantitative microscope here, so it's not, you're a believer when you use real statistics and microscopy and pattern recognition. What we find is that actually there is a clear increase in the amount of laminin binding as we go from the least severe, the most severe to the least severe, okay? But they all, when the drug is administered to their cells, they're all coming much closer back to what we'd consider wild-type laminin binding staining. So, as I said, HUD was asking for an example. Here was an example that we were looking to really develop a platform here. This is not really a talk necessarily about dystroglycanopathies. It's about if you have a dysfunctional cell type, could you think of doing this? And we would love to work with people who had a passion to do that. We have the systems, we have the stem cell capability, we have the high content microscopy, the brains are needed around developing the assay, designing the assay to have a relevant outcome or a phenotype. The next area we move to is really trying to, some of these diseases involve muscle, eye, brain, involve brain. We really wanted to see whether we'd have an effect on neurons. So to have an effect on neurons, you really need to be developing neuronals from the stem cells. So rather than going into all the different forms of CMD, but we moved on to really look at some of these, particularly the defects, rather than, I just showed you the defect here in FKRP. My little mouse isn't working very well. Okay, next we had to look here and say, well, so those were all different mutations in the same protein, the ones I just showed, that panel of 5. What happens if we move to a different mutation in a different enzyme, but in the same pathway, right? Well, the assay still works. So over here was the initial assay I showed you from the FKRP patient. But here, where the defect is in the POMTI1, there's a small increase, but not a very big increase, right? So we maybe need a different assay. We need to look in different cells. There's something, that's not the screen we did. If you remember, the screen we did was screening for this in this patient. So now we need to screen this patient and see whether we could find something that could help them. We also, because in this particular case, this patient has a CNS involvement, we were keen to then move on to generate some neurons. And so following the approach of taking skin cells, making stem cells, and then from those, you can make all these different wonderful cultures of neurons, either mixed cultures, pure cultures. And we've done that. Anna's now moved on to do that, taking undifferentiated stem cells and now coming out, making neurons, glia, et cetera, et cetera. Here just shows you, just give you one last slide really on this, which is, these are the patients, one patient with the POMTI1 mutation here, one of the enzymes that's in that cascade. Here is the child, and here is the unaffected mother. And I think you can clearly see out the far side here, these are neural epithelia, so on the way to neurons. We are getting to neurons right now. It takes 2 more weeks to get out there, and he's cooking them up right now. But I think you can clearly see that the concept of this is going to work just fine. So here you've got the stem cell derived neural epithelia from the child. You can see they don't bind laminin very well, whereas the mother is binding it much better. Again, we have a screen, right, to be able to do it now in neurons. Well, I believe these are neural epithelia, but we'll go to neurons, and I am sure it will be able to do that. So the slide I want to leave you with then, it really goes back to this one, which is really a way of thinking about this, a platform where the genetics will be identified, we know what the gene is that's wrong, we then have to devise the cell-based screen. Having devised the cell-based screen, we put the compounds on individually or in combination, we haven't got into that yet, onto the cell type where we believe the dysfunction manifests itself, then be able to provide that information back as new treatment options. So it's, as I say, this is something we're trying to pioneer here within the Institute, and happy to work with anyone who would be sharing that vision to try and attempt to do this for some of the diseases. So I'd be happy to take any questions if anyone have any.
We've got time for a few questions. All right.
Which cell types did you use for the screen?
Right, for the actual screen there, it was fibroblasts, and we were very surprised that actually we would see a phenotype in fibroblasts, but that's was...
Dystroglycan different glycosylated in fibroblasts, and patients do not have fibroblasts, or they have a muscle
Right,
type.
right.
So I believe the O-glycosylation is different. Fibroblasts.
No, well, this is O-monosyl sugar, but it may be different in there. We've gone on to show that in myoblasts, Anne can speak, she's the expert, but we have gone on to look in the patient's myoblasts and muscles, I mean, and still see we can take the myoblasts, treat them with the drug, and alter the endpoint. There's a lot of things need to be done. You're absolutely right. This is work in progress. None of this is published. It's work in progress to try and get us to believe that whether this approach is going to work. You know, is this approach worth attempting? All right, so that's kind of where it is. I think if we had done the same experiment that I think Hudley's with his fibroblasts and manos, you know, you would find something that would move the needle. He came at it from an intellectual point perspective. Here we're coming at it from a random screening perspective, right? And, you know, how the things work or don't work and whether they're going to work, it just introduces us to a new way, because we don't have many treatment options, right? And we're increasingly have more and more patients where we know the genetic defect. We can make cells, and we can find, in many cases, that those defects are cell autonomous and result in a defect within those cells. And that's not that far away from, if you get a compound that affects that defect of what happens in a patient, to be honest with you.
Yes.
You might talk a little bit about that very effective and I think their therapy here to increase the expression of large to help those infecting this kind of the compound that you find.
Can you repeat the question?
Yeah, so the question is, does it make any sense from the mechanism of action of the 10 compounds that we identified? Not a lot. I can talk to you later about it, but you know, this is the challenge with doing this. It maybe does, and you know, I think that's one of the challenges of doing this, that the first thing scientists want to do is to dig into it and say, why does it work? I just remind you that many of the drugs that were discovered in the 50s, 60s, and 70s, we had no idea how they worked, and we still don't, actually.
Jeff,
Just a quick question.
Jeff, go ahead.
Because dystroglycan, of course, is heavily glycosylated. So if you just look to see if you're actually correcting the glycosylation.
Yeah. So there is definitely an upregulation of the of the O-monosyl. Yeah, there is definitely but I don't know if that's the only reason. A lot of controls need to be done to satisfy someone like you.
He's heard about you.
No, I didn't mean a derogatory way. I just meant. And me, sorry. I know, everybody.
Your type.
Yeah, I didn't mean it.
Phil, just last question here.
Please someone correct me if I'm wrong, but is laminin one of the proteins that associates on?
No, that's lamin A. Lamin, not laminin, right? In fact, there's an interesting side. I know we have to go because of break, but we actually got some samples from UCSD. Someone said, we believe this person has limb-girdle disease. Could you do your staining? We did the staining of it. They had perfectly normal laminin staining, but the quantitative microscope said, hey, every single nucleus in that person's sample is abnormally shaped. With a Z factor was incredible, right? So it says everyone is the wrong shape. And then 2 weeks later, they said, yeah, we've just done the genetics. It's got a defect in lamin A.
Okay. Michael, thanks very much.